Hello, everybody. Welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. I am Francois Franceschi. I'm moderating today's webinar. Revive is a GARDP program to support and connect the antimicrobial research and development community. Revive aims to facilitate learning, to connect people, and to share knowledge. This webinar series is part of Revive's educational activities. In addition to our webinars, we also organize sessions at key conferences in the field. Another aspect of Revive is our expert network. It is one of Revive's goals to help researchers who are not yet well connected to get in touch with leading experts in the field. You can do so via the Talk to an Expert interface on the Revive website. Today, we have two short presentations, one by Eakin and the other by Dan Pevear, which will introduce you to the support mechanisms that are offered by the National Institute of Allergy and Infectious Diseases, one of the institutes belonging to the National Institutes of Health in the US. After the presentations, Anne and Dan will be happy to answer your questions. You can submit your questions at any time during the webinar by using the questions window on your webinar control panel as shown in the slide. We will do our best to respond to as many questions as possible in the time we have allocated. Before we start the presentations, I would like to introduce you to our speakers. Anne Eakin is a senior scientific officer in the Office of Biodefense, Research Resources, and Translational Research in the Division of Microbiology and Infectious Diseases, DMID, of NIAID at the NIH. Anne joined NIAID in 2013 as a therapeutics development specialist as part of the Concept Acceleration Program, where she's involved in efforts to advance promising new therapies, vaccines, and diagnostics targeting all infectious diseases of priority to NIAID, except for HIV/AIDS, which is handled by a different division. In her current role at NIAID, Anne aims to leverage resources and product development expertise to help external scientists advance their scientific discoveries into the clinical development pipeline as novel therapeutics for infectious diseases. Prior to joining NIAID, Anne was a principal scientist at AstraZeneca and an assistant professor at the University of North Carolina at Chapel Hill. Dan Pevear is the Senior Vice President of Biology and Grant Development at Venatorx Pharmaceuticals, a biotech company developing novel antibacterials, which he co-founded in 2010 with Christopher Burns and Luigi Cherry. Dan has over 30 years of experience in both biotech and big pharma, with stints at Sterling Winthrop, Biopharm, where he also was a co-founder, Progenics, Novartis, and Protest Pharmaceuticals. Now, I would like to welcome Anne Eakin. Anne, you can start your presentation now.
Great, thanks so much, Francois, and really thank you for the invitation to participate in this GARDP Revive webinar. I appreciate the opportunity to tell listeners and viewers about the various resources that are available through the National Institutes of Health and NIAID in particular, that we hope will be there to help facilitate discovery and development of anti-infective products. So just to orient people, as Francois mentioned, the National Institute of Allergy and Infectious Diseases, where I'm working, is one of 20 institutes that make up the National Institutes of Health, which is the US medical research agency. And specifically, the mission of NIAID is to conduct and support basic and applied research to better understand, treat, and prevent infectious, immunologic, and allergic diseases. And The division where I work, the Division of Microbiology and Infectious Diseases, or DMID, its mission is to support the extramural research from basic through applied research, aiming to control and prevent diseases that are caused by all human infectious agents except for HIV. And as Francois mentioned, that's because there's a division of AIDS that is focused on that particular pathogen. So one point just wanted to bring up is a unique feature of NIAID's mission is that we have a dual mandate. So not only as other institutes at NIH, not only do we need to maintain and build a robust portfolio of basic and applied research through funding and other interactions, but also we need to be prepared to respond rapidly to any new infectious disease threats that come up. Things from Ebola outbreaks to Zika outbreaks across the globe, as well as the emerging antimicrobial resistance. And as I mentioned, DMID is responsible for pretty much all pathogens of interest to human health. And so that is a responsibility that spans over more than 300 pathogens. And you can get an idea of the budget and the amount of investment that the US is putting in that space with the fiscal year 18 budget listed there at about $1.7 billion. So one of the main focuses of NIAID is putting forward support mechanisms, various support mechanisms to reduce the overall risk of product development and encourage and advance new efforts in this area. And so I want to take you today through the different mechanisms that are available for all researchers. And I just want to bring up the point that while the NIAID is US based, obviously part of NIH, all but just one small part, and I'll highlight that, of the things I'll talk about today are open for application regardless of where you are in the world. So just going through some of the various mechanisms that are available, I think, you know, grants and investigator-initiated grants are kind of well known, or that's what NIAID is, or NIH is well known for. It's really the foundation of how we fund basic and applied research. But there are a couple other mechanisms I wanted to highlight as well. One is product development contracts that are funds that are given out to companies specifically in the area of translational research and research toward R&D and developing new products. And then finally, we have not direct funds to investigators, but we set up research service contracts with different service providers, and we make those services available to external scientists for determining key assays and providing key data that may help push their programs forward. And so basically this is the outline of what the slides I want to go through next, really highlighting those three funding mechanisms that we offer product developers in this space. So starting off with the grants area, as I said, I think this is the space where NIH is most well known for, and that's the bulk of the funding going to investigator-initiated awards. Your typical traditional R01 awards are also smaller R21 focused awards. And also there are small business awards, and this is the one and only example that is going to be only open to US companies, and that's the small business awards. All the other grants and contracts that I'll mention are open to application regardless of where you are globally. And so these investigator-initiated examples of grants have standing submission deadlines that come 3 times a year and really are a grassroots way of funding ideas and efforts out in the scientific community. So in addition to those standing grant opportunities, NIAID will also put out specific focused grant requests. So one area that's commonly leveraged is called the Partnerships Program. And that's where we'll put out periodically, we'll put out specific requests that target a particular research area. Often that's focused on translational research and product development, but also it can sometimes be to bolster efforts in a particular basic research topic. And so the best way to stay in touch with these opportunities for new grant announcements that come out is to follow online our funding opportunities, and I've listed a website there. And also the best bet is to subscribe to regular email alerts through NIAID funding news. So the regular submissions for the investigator-initiated awards, that comes up on a regular schedule that you can be prepared for, but these focused awards come up just from time to time and aren't on a regular schedule. So another thing I wanted to highlight to people is the best way of getting the earliest view of potential upcoming grant announcements and contract announcements is to pay attention to what we call our NIAID Council cleared concepts. So we have a NIAID Council that reviews all of our grant and funding actions, and they meet on a regular basis, about quarterly. And what comes out of those discussions will often be initiatives or concepts that NIAID internal staff are proposing to potentially turn into a new funding opportunity. And so your first view of things that have been approved by our council that are likely, although not guaranteed, they're likely to become a new announcement will show up on that NIAID Council cleared concept page. So I definitely encourage you to keep an eye on that as well. So in terms of current opportunities that are specific to the antimicrobial resistance space, right now there are 2 program announcements that are out on the website, and these are focused on generating new insights and mechanistic understanding into antibiotic resistance development. And particularly what those program announcements are calling out are encouraging people to send in applications for grants in the area of microbiome involvement in drug-resistant infections, as well as any efforts to understand interactions between human microbial communities and drug-resistant bacteria. And a third topic is in terms of better understanding and characterizing new combinations of existing drugs with the aim of addressing and treating infections caused by MDR gram-negative bacteria. And so what these program announcements are intending to do is encourage people working in this space to submit investigator-initiated grants to the regular NIAID submission deadlines, but it's just highlighting a few areas that we're interested in receiving applications. And then again, this is US focused, but for anyone who's in a United States Small Business Administration approved company, there are SBIR and STTR grants that are open to you. And these are really good grants for helping kickstart some early exploratory work in the phase 1 through more focused research and development efforts in phase 2 of the awards. And one of the things I wanted to highlight, again, these come up with regular submission dates 3 times during the year. And they're open, as I said, only to US-based Small Business Administration approved companies. But one of the things I wanted to highlight particularly that may not be well understood is there are particular what are called waiver topics that allow you to extend the base time period as well as funding limits quite substantially. So that's something to absolutely look into. And our waiver topic for DMID is very broadly defined, and basically AMR is a high priority for the division, and so people working in that space of product development will definitely qualify as a waiver topic. And so the last thing in the grant space I wanted to highlight is we currently have a request for information, and that's particularly in the area of antibacterial resistance research. So in 2014 was the last time NIAID put forward the strategic plan for addressing antibacterial resistance, and we're now refreshing that plan. And one of the important components of that is reaching out to the external scientific community to ask for comments and suggestions on what you all see as key strategic areas and approaches that need more work. And so we definitely would request anybody, regardless of where you are, to please weigh in, give us your suggestions and comments. And there's a web link which has information on how you can submit those ideas. And the deadline's coming up pretty soon for that, so we definitely would appreciate your input there. So now moving on to the second type of funding mechanism, and that's our product development contracts. And so these are contracts that are focused on supporting the development of new products targeting infectious diseases, either therapeutics, vaccines, or diagnostic products. And these are contracts that are given directly to companies who are working in this space and are up to about 5 years typically, and they're milestone based where options can be exercised to add to the funding as the project progresses along during that time. And the announcements for these product development contracts come out as requests for proposals or broad agency announcements that appear on Fed Biz Ops, which is basically all things federal government directed in terms of contracts. And the awards are typically in the range of 3 to 40 million, depending on the scope, and can be, quite beneficial for driving forward programs in this area. Again, you'll get highlights of new product development announcements will come out of the Council Cleared site that I mentioned before, as well as in the NIAID funding news. And so one point I'd just like to highlight about these contracts are not only are they a good source of funding for your efforts, but also we set them up as a pretty close partnership with NIAID staff. And so we form, along with your cross-functional project team at the company, there'll be DMID staff of various backgrounds and expertise that join that effort to meet regularly and discuss, how best to drive the program forward. And so we, hope that this decreases the overall development risk for the project, not just through funding, but also through contribution of expertise, and can help fill in gaps perhaps that smaller companies may have in expertise. Currently, we have in our portfolio of product development contracts 11 vaccine contracts, 9 therapeutics efforts, and 1 diagnostic effort going on at the moment. And again, these come up approximately annually with new announcements going out. And just to give you an idea, over the last 10 years, there are here just listing all the various companies that have been supported through either vaccine, diagnostic, or therapeutics development efforts and various programs. And so you can see, the support ranges from smaller academic spinouts to larger pharma and biotech companies. So on to the third area of the funding mechanisms I wanted to highlight today are the NIAID preclinical services. And so the goals of these services are, unlike the prior 2 examples of grants and contracts, these aren't sending funds directly to investigators, but instead are providing a mechanism for external scientists to request specific gap-filling assays or key data that is needed to de-risk or drive their products forward. And so our hope there is that by providing access to specific assays and services that might address key go, no-go decisions or key data that may be needed as, say, preliminary evidence for grant or contract submissions, things like that, providing that key data can help push the programs forward. And this not only provides the specific assays, but we also provide expertise in product development that we can help design the experiments that are needed, as well as provide access to contractors who can provide the data. And importantly, we set up these contracts such that no loss of intellectual property occurs and everything is maintained, all your data and information about your program is maintained in strict confidence going forward. So the way we have the preclinical services set up is NIAID sets up in advance suites of contracts that provide a broad range of different assays and capabilities. And so NIAID establishes these relationships with contractors and service providers, and then we provide and pay for those services to be conducted, and external scientists can ask to access those particular assays to help drive their programs forward. So the next few slides, I'll just take you through some specific examples of what those various assays and various contracts have to offer that you can then consider to see if they might help with your efforts. So one of the contracts is our therapeutics development services that offer in vitro testing and assessment of antimicrobial activity. So the contract that we have in place is very broad. As you can imagine, we have to cover the possibility of interest in many different pathogens of interest to the division, but we have these divided up into the various contractors and a pool of contractors that can offer these various broad capabilities. And this offers focused screening as well as more medium to high throughput. And specifically in the area of antibacterial testing, we offer MIC testing and MIC 90 against a panel of clinical isolates of a variety of pathogens, variety of bacterial pathogens with different susceptibility profiles. That can be done as monotherapy or combination therapy, depending on your program needs. And we leverage the FDA CDC AR bank that is available, which provides very current clinical isolates with interesting drug susceptibility profiles that you may be interested in profiling your new products against, as well as other isolates that are available in the contractors' labs. And another area is biodefense pathogens. So not only do we offer the pathogens of interest to public health, the typical CDC and WHO threat list pathogens, but we also offer biodefense pathogen testing. And so that data is often useful to help you set up for getting grants or future contracts through other agencies that may be focused on biodefense. So another contract we have is, again, this is through a pool of different contractors that bring capabilities of testing in various models, animal models of infectious diseases, or in some cases in vitro models of infectious diseases. And these models can be used to provide early efficacy evidence as well as more detailed PK/PD of novel therapeutic candidates, as well as testing of vaccine candidates as well. And so particularly for bacterial infections, we offer currently murine thigh and lung infection models, as well as UTI models in diabetic and normal mice. And we have a hamster model of Clostridium difficile infections. And those are just contracts that we use fairly routinely and frequently for bacterial projects. But we also have the ability to set up novel models as needed. And so if you had a specific animal model you were interested in, you could come to us and talk about the possibility of setting that up. So another large contract that we have set up with a handful of contractors available are for the general therapeutics profiling. And that includes both chemical synthesis as well as ADME profiling and toxicology profiling of candidate therapeutics. And this can include both medicinal chemistry analog work as well as scale up, even including GMP manufacturing of new products. Also in vitro and in vivo, preclinical tox and PK, including those studies that may be needed for regulatory authority submissions, such as an IND. Those GLP tox studies that are required for that, we can support. One gap that we noticed is for early sort of lead optimization, where really quick turnaround of data is important. So we've established a rapid screening contract for ADME profiling and PK screening for that particular phase of discovery and development of new candidate therapeutics. And these contract services can also help provide overall planning and evaluation of strategies for products, as well as IND documentation as needed. And then in terms of larger, more biotherapeutics, we have a separate suite of contracts that are in place for supporting biotech-derived or biotech-focused products, such as monoclonal antibodies, recombinant proteins and peptides, or nucleic acid-based products. And so these contracts can do not only overall product development planning and evaluation, but can also do manufacturing and assay development for product testing and release. And also the contract is set up to be able to access and provide reagents for diagnostics product development, as well as providing regulatory documentation for that manufacturing. In the area of vaccine development, we have two large contracts. One is focused on manufacturing services, and that can do anything from early feasibility testing to larger scale manufacturing and product release assays, and can also include the regulatory documentation needed for those larger scale manufacturing. And the other vaccine contract is focused more on testing, and that includes assay development for in vitro assessment of products, as well as animal model work that's needed, including for those pathogens requiring animal rule pathway work, including the biodefense focused, as well as emerging infectious disease outbreak type pathogens. So finally, just a couple words on eligibility, just to make it clear that these services are available to you regardless of the type of organization you're from, whether it's academic, nonprofits, industry, government, whether you're located in the US or anywhere internationally, these services are eligible. You are free to apply. You do not have to have funding or current funding or prior funding from NIH to apply for these. And it's a fairly simplified request process. Requests are short applications that can come in year-round and are reviewed internally for approval. And really the things that we're looking for in terms of approving requests are the priority of the research area that you're working in, and the significance of the program you're trying to push forward, as well as importantly the preliminary data and your justification for what assays you need and why. And a key part of that evaluation is your overall product development plan and how the data you're requesting will fit in to that overall plan. So briefly, in addition to the preclinical services that I went into in more detail, I just want to make sure you're aware that we also have contracts available to support clinical studies. And again, just like the preclinical contracts, these are run as established contracts that NIAID pays for, that are in place and there to help with early clinical trials of promising vaccine, therapeutics, or diagnostics. And we have both phase 1 clinical trial units that are focused mainly on phase 1 studies for therapeutics. And then we also have vaccine and treatment evaluation units, which are several contracts lined up for supporting clinical trials, including patient trials as well. And also in the area of a clinical trial or clinical network, we have the Antibacterial Resistance Leadership Group, which is funded by NIAID. And the purpose there of that group is to focus on antibacterial resistance and prioritizing and designing clinical research that will help reduce the threat of AMR. And there are many different areas that this group focuses in, and I've listed the website there, as well as a clinical infectious diseases issue that focused on the various activities of this group. And they've been quite active in the space, including both therapeutic studies as well as diagnostic studies. So finally, the last thing I want to bring up today is NIAID's involvement in the CARB-X program. So I think you all are probably mostly aware about the CARB-X program. That's a global partnership that's set up and run out of Boston University that maintains a portfolio of programs all focused on developing new interventions for antibacterial or antibiotic-resistant bacterial infections. And NIAID participates as an original funder, and we provide in-kind services, or in-kind support, I should say, that leverage our preclinical services that I just went through to help support those programs, as well as technical expertise that we offer companies involved in CARB-X. And we've committed up to $50 million of in-kind support over the 5-year period that CARB-X has been awarded. And this slide is already a little out of date. It changes very rapidly, but of the 36 CARB-X programs, including 2 graduating projects, NIAID was involved previously in several of those, a little more than half of those programs, with either grant support or contract support or PCS in the past. And since the programs have joined the CARB-X portfolio, we've supported 22 of those projects and in excess of 100 or more studies, and we continue to do so going forward. So that's all for the overview that I had. I would just like to finally point out that not only am I happy to answer any questions that come up today, but also I've listed my email address there, as well as a couple of web links. If you have any questions in the future, I'm happy to help address those.
Thank you, Anne, for the very clear presentation. And I remind our listeners that you can ask questions at any time during the webinar using the window that you have in your webinar app. And now Dan Pevear will give us some real-life examples where NIAID has helped by providing services to Venatorx. Welcome, Dan.
Thank you, Francois. Can you hear me?
Yes, we can hear you.
Okay, great, thank you. My name is Dan Pevear. I'm the Senior Vice President of Biology and Grants Development here at Venatorx Pharmaceuticals. I am joined by 2 of my colleagues, Dr. Dennis Daigle, who is the Director of Biology, and Dr. Stephen Boyd, Director of Chemistry, who have actually been the individuals with hands-on experience with the preclinical services. So I was asked to say a couple of words about Venatorx. We were founded in 2010, as Francois mentioned, by myself and Chris Burns and Luigi Xeri. We had 3 employees. We're now up to about 60 employees. We're located in Southeast Pennsylvania, near Philadelphia in the United States. We have about 25,000 square feet of office and lab space here. We are a fully integrated discovery and development organization, privately held. We have been very, very fortunate to be able to take advantage of the push incentives that have been offered largely by the NIH, but also through Wellcome Trust and DOD and CARB-X, and have been awarded more than $120 million from these organizations in order to develop our portfolio. We have actually been working now with Anne and the group at NIAID and DMID for the better part of this decade, and I'll just add that they have been amazing collaborators and have provided a tremendous support group to us. And then one more self-promotion slide. This is the Venatorx pipeline as it currently exists. You will notice that in red are all the ways in which NIAID and other organizations have and the funding has been leveraged in order to promote these programs. So our lead program is an IV injectable product, cefepime with Venatorx 5133, a beta-lactamase inhibitor. So this is an ultra broad spectrum beta-lactamase inhibitor that covers both serine and metallo-beta-lactamases. It is currently the subject of an NIAID, one of the product development contracts that Anne mentioned, as well as an award from the Wellcome Trust. Following that is a beta-lactam with an oral beta-lactamase inhibitor combination product that covers both ESBL and carbapenem-resistant Enterobacter ACA infections. This was discovered through NIH grants, an R01 and an SBIR, and then ultimately was awarded another product development contract from NIAID. Again, on both of these contracts, we have been working with Anne and her team very closely. We also have called a PVP inhibitor program. So there's a new class of antibiotics that target penicillin binding proteins, but they're not susceptible to beta-lactamases, so they're not a beta-lactam-based compound series. This is funded by both CARB-X and DoD, and it's specifically in this program with CARB-X funding that that Dennis and Steve are going to be talking to you about today in terms of our use of the NIAID preclinical services. And we also have programs in antivirals where we have some very, very interesting work going on in both hepatitis B and in chikungunya virus that's supported by the NIH. Okay, so we have all of the different funding mechanisms that Anne mentioned. We have leveraged these extensively to establish the pipeline and to move our compounds into early development. The preclinical services opportunities, I think they're a wonderful opportunities that frankly we didn't use that much when we were starting out because we weren't that familiar with this mechanism at NIAID. But I think there are places where, you know, if we knew then what we know today, where I see that there's some real value to the PCS. And the key to all of this, the key to being successful with this mechanism is that you need to plan ahead. There's a process involved. There's time to set up the contracts. You know, there's paperwork to do. There's a pre-approval process. Once you go through all of that and actually ship compounds, what you'll see is that things move along pretty quickly, but there's some lead time. So if you're a startup looking to generate critical data on a compound series, maybe for an investor pitch or for a grant or contract submissions, perhaps you're an established company like ours, but you are looking to do a portfolio expansion. So you have project number 5 is unfunded within your portfolio, and you'd like to generate some preliminary data, but not actually, you don't actually have support internally to fund that work. Perhaps you have submitted a grant application, gotten your summary statement back, didn't get quite the score that you wanted, and you want to prepare for a resubmission, and you need to generate some new data. And as Anne also mentioned, I think one of the things that the PCS gives you is it gives you access perhaps to the subject matter experts, so various forms of support. I think all of these opportunities, all of these different activities are perfectly suited for the PCS. If it is something, I think Anne would be the first to admit that if it is something that is on critical path and is extremely time sensitive, this is not going to be the fastest mechanism. So again, I emphasize plan ahead. If you plan ahead, if you storyboard what you need to do and begin this process early enough, this can be a very effective mechanism of which to advance programs. So that I wanted to actually finish by presenting 3 real-life examples. Anne had asked us to do this, and 2 of them are on more the ADME type activities within PCS, and the third one is on a chemistry synthetic effort that is ongoing. And this is all through the CARB-X accelerator, as I mentioned. And what I want to do is give Dennis and Steve a little bit of airtime here so they can give you their firsthand experience on how this has gone. So I'm going to now turn this over to Dennis Fagan.
Great, thank you, Dan. So first off, I just want to say I'm glad that I have the opportunity to help Anne and the NIAID PCS team to get the word out on the available services and the advantages of these services. Do keep in mind that this program that we're about to describe, or the 2 projects that we're about to describe, have already been pre-approved by NIAID. They are and do also have an acceleration due to the fact that they're CARB-X funded. So essentially, the 2 first that you see on the next 2 slides are essentially in vitro ADME, enzyme selectivity, and plasma stability that, again, are not critical path at the time that we sent these submissions out. And the critical dates or timelines that I think most people are concerned with are data readout. I would say that our impression of the services as we've gone through over the past couple of years are that the only delay that you see is on the front end, which is getting everything approved internally. Once you ship compound out for studies, typically the data readout is within 4 to 5 weeks of compound shipping, or 10 weeks from beginning of initiation of the service request forms, which is, you know, it's a bit longer than usual, but once the compounds go out, you're essentially on the time frames of the selected vendors that NIAID has put in place, which would be essentially equivalent to what you would have if you were to externally or initiate those studies by yourself. So the second portion here, and I want to highlight, I mean, there's not much difference between the first and second projects. Similar analyses were requested, similar timelines were achieved, again, compounds being shipped out 6 weeks post service request form approved, with a data readout 4 weeks later. So again, it's pretty fast once you have submitted the compounds. What I would like to say, though, is that I would say that, of course, these vendors that are selected by NIAID are professional organizations. The data that we've received from these services have been top-notch, and also, admittedly, the interactions that we've had with NIAID staff to coordinate these activities have been great as well. So I'm gonna pass now to Steve Boyd, our Senior Director of Chemistry Research, to talk about the last example.
Hello, and thanks to Dan for allowing me to at least chime in here in support of the preclinical services chemistry piece. This is something that we haven't previously taken advantage of, and so this is our first foray into this area. But we were very pleased to learn that part of this suite of services included chemical synthesis. We do have a internal chemistry team that's responsible for all the SAR development work that we've done on the programs, but we occasionally have need for large-scale synthesis of, in this case, a comparator molecule that we needed to have prepared so that we can benchmark our biological studies that we intend to do for the program. This particular compound is actually very complex synthesis, and so for us to take that on and to do it internally would definitely divert from the chemistry resource that we have. We have a relatively small team, and so being able to leverage this capability externally really saves us time and money to be able to perform that. So we started out by testing the water shear to figure out what was required in order to do this. Found out yes, we are able to do this through the NIAID preclinical services. So we've submitted the concept, and the synthesis has actually been initiated, and the timeline for that is pretty much on par with what we would gather from talking to other contract synthesis groups to kind of get similar quotes on their time frame. So we expect to have the compound in time for us to do some very key studies to support the program going forward. So far we don't have any final answer for you on this, but it's moving forward.
Yeah, so that's this is Dan again. So I think that's really all that we wanted to say. We've used this mechanism. We think it works great. You just need to plan ahead a little bit on it. And there's a broad suite of applicability here that I think we would just encourage people to look at if this fits with their timelines and take full advantage of this opportunity.
Okay, thank you, Anne, Dan, Dennis, and Steve for the interesting presentations. We will now start the Q&A session. We have already received some questions. Please keep sending your questions via the question window, and we will do our best to answer as many questions. So I have here a couple of questions, so I'm going to start. The first question says, do research tools used in the discovery and development of antimicrobials qualify for SBIR and other grant opportunities with NIAID? for example, a novel assay technology or a high throughput screening technology, or is just vaccines, therapeutics, and diagnostics?
Right. Thank you for that question. So I think that for the most part, our direct-to-you contracts are focused on supporting, as you said, the therapeutics, vaccine, and diagnostic products and candidate products. That said, I think SBIRs are more broadly open and are open to what will drive your business forward. And so I think you could make the case for that sort of assay technology development to fit within the SBIR grant programs. And then also, should those efforts advance, I think, and to the point where they would be useful to include in the NIAID preclinical services. It doesn't come up very often. We don't compete those large service contracts, but every several years. But keep your eye out if you do have a particular assay or technology or capability. Excuse me, we do solicit for contractors to join those pools of contracts that make up the preclinical services. So that would be another avenue to get funding and support and involved.
Thank you, Anne. Then we have several questions that are related, and I'm going to try to kind of merge them into a single one. In this sense, the first one says, is it possible to get a list of the strains in the available MIC panels? And somebody in relation to this also asked, let me just get to that question because it was later. Essentially was saying, are these all well-characterized resistance mechanism defined strains? And if they are also recent isolates less than 3 years in alignment with regulatory guidances?
Yep, thank you. Great questions. So I guess a couple of things. I mean, one, certainly when we are starting to talk about specific studies, we can get more details out to you. We tend to not put up a website that lists out strains because that kind of thing changes a lot. But one thing we are trying to do is to leverage the AR bank that's being set up by the CDC and FDA. So if you could go to their website, just search, antibacterial resistance strain bank, FDA, CDC, and you'll find it. And that site has a lot of information. Most of those are recent clinical isolates, and most of those strains or isolates have been fully characterized, and that information is posted on that website in terms of phenotype, drug susceptibility profile, as well as links to gene bank where you can examine the genotypes. And so our contractors that are doing the MIC testing use those strains as well as other strains that they have internally. And so, as I said, if there are more details needed as the studies being planned, we can have you communicate with the contractor. And that's a point I probably should have made more clearly before. Any of our preclinical services, it's not like we're taking your compound away from you and running studies that we set up with the contractor, but instead you are directly involved in the planning of those studies and the design of how they're conducted, and interacting with NIAID and the contractor in doing so. And so details around strains could be pulled in through those discussions as well. But a good place to start is to look at the FDA CDC strain bank. And if I could add one more thing to that, not only do we leverage those strains or those isolates for the MIC testing, but we also are trying to provide many of those strains in animal models. So subsequent in vivo efficacy and PK/PD testing can utilize some of those strains as well.
Thank you, Anne. I have another question here. It says, is the NIH planning to reinstitute their therapeutic testing in murine models, for example, TIDE, lung, PK, MTD program, as they have done in the previous 2 years, or is it expanding the sites performing the preclinical services?
Right. So the sites that do the preclinical services are always changing. So we have pools of contractors, and that given who the particular contractor is that's on board for running those assays, that can change. And so that is specifically to the area of the mouse infection models, the thigh and lung models. We are definitely trying to build capability in that space. As I mentioned, leveraging the FDA CDC strains is one area. In addition to sort of leveraging the broader capabilities to include PK/PD as well as efficacy testing. So for instance, the mouse thigh and lung model contracts we have in place include pharmacokinetics in infected mice so that we can do more PK/PD analysis. And I should just emphasize across the board for all the preclinical services, while those are being services are being provided by contractors, NIAID pays for those contract fees. And so the assays, once you submit an application and get approval, you receive those assays and services free of charge to you. So NIAID covers those charges. You are responsible for just submitting your compounds to the contractor. You get the data back to use as you see fit going forward. So the data is yours to use, but the services are free of charge.
Thank you, Anne. There is also here another question. Is there a computational model service for lead optimization available through preclinical services at NIAID?
Right, so I guess you mean in the area of medicinal chemistry analog design. I'm assuming that's the question. So yes, I think there's maybe not formally set up as sort of an automated type approach, but definitely I think we could pull in our contractor who does the medicinal chemistry to help with the design and feasibility of different analog work. I think it helps things run more smoothly the more experience you have internally with synthesis of the compounds, but certainly with providing some back and forth advice of specific analogs, that work could be included as part of that kind of a study.
Thanks, Anne. I have here another question asking if you could comment on NIAID's commercial readiness program.
Right, so there's a subset of the SBIR program, I think is what you're referring to. So companies who have SBIR funding at phase 2 have available to them the commercialization readiness plan program, I guess. And I think that is, for a while, it was taken off the books. And as I understand it, they are just setting up, excuse me, for the fall timing to have that reinstituted. I think the exact details of what that's going to entail are still left to be finalized for the fall announcement. My understanding is that it focuses on some of the, excuse me, some of the extended, more expensive efforts that go toward later stage product development. And so you can start getting a bit higher budget items that lead toward commercialization, larger scale manufacturing, that kind of support. Sorry, I don't have more details to tell you, but they should be later this summer putting out descriptions of exactly what that's going to entail when it hits the street again in September time or October timing.
Thanks, Anne. I have here a question asking if you could comment on how many R01s go to companies and if any overseas companies normally receive R01s.
Yeah, that's a good question, and I'm sorry I do not have the statistics on that, so I can't give you firm numbers or percentages. I can definitely tell you with confidence that the R01 mechanisms for grants are open, and there are awards given to non-US based companies and institutions. I apologize, though, I can't tell you exactly what the numbers break down.
Okay, here I have another questions that maybe was partially answered in Dan's, Dennis and Steve seminar, but maybe you can comment in general lines. There is somebody asking, what is the turnaround time after submitting a request to NIAID preclinical service until you receive a report on the study?
Oh, okay. Well, actually, the two examples I think that we listed on those two slides actually had the final reports. So typically we saw 10 weeks from initiation of SRF to actual data readout, and then typically it was 2 to 5 weeks for final report status afterwards.
Thank you. There is somebody else here asking, if a company that wants to access NIAID preclinical service but is not funded by CARB-X, is the process any different than the process for CARB-X funded companies? I mean, that person also was asking for a price list, but as Anne already pointed out, all those services are free of charge. So.
Right. Thank you for reinforcing that. So yeah, the overall process is very similar, regardless of whether you are a CARB-X company or whether you're outside or independent of CARB-X. So our preclinical services are open and available to everyone, regardless of whether they're in CARB-X or not. The advantages you do get as a CARB-X awardee is some of the front-end review processes and submission processes have been streamlined due to the prior vetting, if you will, of the program and the fit in terms of priority with NIAID. And because we've been involved with that goes a little bit more streamlined and a little more quickly than if you were coming in de novo to describe your project to us. But otherwise, the rest of the steps are very similar, whether you're in CARB-X or outside of the CARB-X program.
Thanks, Anne. Here, there is another question saying, once the first task has been approved by NIAID on a given project, will approval of subsequent tasks be faster or possible?
Yep. So absolutely. It's not a one request and done type situation. So we are open to discussing subsequent tasks and requests going forward. And what it kind of depends on exactly what the scope is, but at least in my experience, what helps is that in the initial conversations you have with NIAID staff about what your needs are, if there is some obvious steps in the process, I think it's really useful to sort of describe that up front. So if you're interested in, for example, having your therapeutic candidate tested in in vitro ADME with the idea that you wanted to test 2 compounds in vitro ADME and have a PK on the selected compound based on that in vitro data, we can kind of program in the idea up front that given promising data, we could probably approve both requests. So I think the short answer is yes, we do accommodate more than one study and can review requests for more than one. It just helps if we are sort of discussing that up front, and so people are aware going into it what your big picture questions are.
Thanks, Anne. I have another question here. Somebody's asking if the research service contracts offered, for example, to support diagnostics can also include proteomics or hardware prototyping, for example.
Yeah, that's a good question. So I'd have to, maybe get into more of the detail. I would say that we are very focused on trying to get our preclinical services more adept at helping diagnostics developers. In the past, we've tried to set up some things, but we find that they are very kind of customized to the various platforms. And so I would definitely invite whoever asked that question to get in touch, and we could talk about details about what's possible. I think I guess the other thing I'd throw out in the space of omics, genomics, proteomics, and that kind of thing in addition to the preclinical services contracts that I mentioned today, NIAID also sponsors centers that do those kind of omics work. So even if we might not have easily available support through our contractors that I talked about today, there may be other NIAID avenues to get that kind of support and interaction. So please, diagnostics developers, let me know what your needs are, and we'll see what can be done.
Thank you, Anne. And the same person who asked that question was asking if you guys are aware of any SBIR-like type of funding available for non-US early stage diagnostic companies, for example.
Good question. I am not. I'm not directly aware. I mean, outside of CARB-X's involvement in diagnostics, I don't have personal familiarity with other non-US based agencies that could offer something like that. Sorry.
Yeah, I agree with you. And CARB-X is probably the best one on that case. And also there are individual initiatives in different countries supporting SMEs. So it will be a matter of depending on the country you are. I have another question here. Somebody again asking for the strains and asking if NIAID can supply strains for internal use. For example, if you have done a study with NIAID and would like to use some of the strains for your internal use, if NIAID will be able to supply those.
Yep, that's a good question. So I guess one of our motivations, I guess a couple points. One of the motivations we had for wanting to leverage the CDC FDA strains is that they are available. I do understand that it is somewhat limited in terms of international accessibility, so that is a gap that we're involved in conversations about how to fill. So definitely in the US, access to those FDA CDC strains are fairly straightforward. Internationally, it may be more difficult. So I think specifically, it would come down to the contractor and their ability to share the strain. I will also throw out that we have a general repository called BEI that has various reagents, including bacterial strains, that are available for ordering regardless of where you are geographically. So the BEI strains that are available can be shipped readily internationally. And so that's another resource that you could look into for obtaining specific strains. But I would just say that if it will depend on the specific strain and the specific contractor, how free they are to ship a strain to you after running an assay. But that kind of a question could be discussed up front.
Thank you, Anne. So somebody's asking, I guess there is a bit of confusion, or it's not clear, if now all the services that were offered directly by NIH and BARDA are now channeled by.
You broke up at the very last there, Francois. Could you say that again?
Sorry, the person was asking if now all the funds that are being channeled to AMR are being, say, distributed via CARB-X, or if still NIAID and BARDA play essential roles on funding these initiatives independently of CARB-X.
Yep, got it. So yeah, thanks for that question. So it's very important. So the CARB-X program is certainly something that NIAID and BARDA participate in through the funding, but that is not the only funding in the space of AMR and antibacterial resistance efforts. So all of the product development contracts I mentioned, all of the grants are really going on as usual and outside and in addition to the efforts going on CARB-X. And that's the case for BARDA as well. They have other funding mechanisms and opportunities in this space beyond just the CARB-X program. So while CARB-X is a new opportunity that's very exciting and that U.S. government and other funders are actively involved in, that by no means is the only way you can get funding for efforts in this space.
Thanks, Anne. Somebody from the CRO side asked, how can a specific CRO with capabilities within the antimicrobial arena be included in the list of companies that are used by NIAID for research service contracts?
Right, great question. So as mentioned, these the contracts that offer the preclinical services, or that make up the preclinical services, they we solicit proposals to participate in that program on a regular basis. That said, we generally have those contracts set up for about 5 to 7 years, and so you have to hit the cycle right to see that kind of announcement go out. So definitely watch the space, but I do appreciate that there's a gap, a long lead time to get involved in the program. So short of that, if you had a specific capability that may be unique that could offer in this space, the contractors who are members of the preclinical services pool are generally publicly known, and so you could always reach out to those contractors and perhaps get into an agreement where you are a subcontractor or something like that during that interim time when the awards are already in action and in place. But yeah, if you watch out for the new announcements and the new solicitations when they do come out, that's the best way to get in to become a member of the pool.
Thanks, Anne. Somebody's asking if NIAID PCS plans to offer any additional types of services in the future, and how those PCS decides when or whether to add new services.
Yeah, definitely. I think we're always looking to build the capabilities, and some of the ways that we learn about what's missing or where the gaps are when we receive questions and requests from the community that we realize that perhaps aren't easily set up. And in cases like that, we try to explore various mechanisms to access those earlier. But in the worst case, the next time the cycle comes around, we would try to add that capability to our contracts going forward. So please don't hesitate if there's something that I did not mention or that you think are important, whether you offer it or whether you need it, to let us know, because that's something that could be added, and we're always looking for new ideas.
Thanks, Anne. We have another question here. So somebody's asking that essentially they are working on some entity that has membranolytic activity and that has very strong activity against biofilms and in gram-positives and gram-negatives, but they are asking, the majority of NIAID grants are focused on something else. So will research be supported that, for example, has to do with cyclical peptides or anything like that, so not small molecules?
Yeah, no, good question. So I think the investigator-initiated grants that go to the regular standing study sections, they're wide open to capturing whatever ideas and projects you have pushing forward. And so I definitely encourage you to keep active in that space and submitting grants. In terms of specific directed focused opportunities, I know we've had a few in the past that are focused on so-called non-traditional approaches that that might fall within that category, and so watch for those opportunities. It's hit and miss because they tend to be more focused. But if there's a particular area that you're working in that you think needs to be elevated for more activity, as I mentioned, we were asking for requests for information and trying to get ideas and suggestions from the community for areas that we should focus our future strategic directions and plans in the AMR area. So please, submit your ideas there, and we'll see if we can get them added and included in the overall strategy going forward.
So somebody else is asking, at what stage in development are therapeutics accepted? So do you have to have already a lead, or you can be in the very early stages? Is there a limitation on that sense?
Right, yes, good question. I think, for the grants, it's wide open to any stage in development. For our preclinical services, they tend to fit best when you have lead, molecules or lead vaccine candidates, just because that it sort of lends itself better to the specific requests for assays. And so the scale and throughput is not such that we fit well with larger scale screening or testing of multiple analogs or hits, and instead we do better fitting in that sort of lead follow-up type assay space. However, the grants are a great place for doing more earlier basic type or early discovery phase efforts.
Thanks, Anne. Somebody's asking if only gram-negative compounds are accepted, or if you, for example, have a novel mode of action anti-MRSA compound, will that be considered?
Absolutely. So I think we are certainly mindful of the threat of gram-negative drug-resistant bacteria and hospital serious infections. That said, MRSA is still a high priority, and so any new intervention approaches you have targeting that pathogen is also of interest to us.
And somebody is asking if testing services include, for example, Mycobacteria and Clostridium difficile.
Right. So the C. difficile, I don't believe we have currently in vitro testing set up. The capability is there. We just don't have a contract in place. So it would, if we were able to set it up, it would be a bit longer lead time, but still perhaps possible. We do have the animal model for CDI set up for testing.
And Mycobacteria? Can you hear me, Anne?
Well, sorry, what was that? I didn't hear that.
They also were asking about Mycobacteria.
Oh, so we do have the tuberculosis we have set up for testing. The contract is just being set up now, so it'll be a bit of a delay, but we have testing services for MTB as well.
Thanks, Anne. Somebody's asking if there is any statistics that you can consult, or if you have any that essentially gives some kind of like success rate of foreign-based applications as opposed to US-based research groups or startups.
I do not have that at my fingertips. I wouldn't be surprised that it may exist. So if you wanted to send me an email, I could try to find out more information. I don't have that in front of me though right now.
There is also somebody asking here, is NIAID will also focus on foodborne pathogens?
Good question. I mean, I think as it relates to causing human disease, I think, yes. I guess if it were just, to protect the food supply in that sort of a sense, I don't think that would be as directly relevant.
Thank you. Somebody's also asking if in the PCS services you offer any kind of mode of action or mechanism of action testing.
Yeah, that's a question that comes up from time to time, and so far we have not gone into the more specialty microbiology type support. It's definitely something we're considering a lot. So, if you had that need, please feel free to bring it to our attention and we'll think about it. So far, we have not gone into the mode of action or, killing kinetics or resistant frequencies, those more specialty tests outside of MICs. I think our contractors might be capable though. So given the right sort of request, we might be able to put something in place, but currently it's not there.
Thank you. There is also somebody asking if you could say a little bit about confidentiality so what is the confidentiality that is granted by the preclinical services of DMID?
Sure. And so before any interactions, excuse me, before any interactions with our contractors for running assays, the first step of the process is to sign what we call our non-clinical evaluation agreement, and basically that's the legal terms agreement that spells out in detail how confidentiality is maintained, how IP is protected, and that kind of thing. And so, I'd be happy to send out that agreement to anyone who wanted to see and get their lawyers to weigh in. But the intent behind that is to maintain strict confidentiality. We enter in and have that confidentiality established between NIAID and our contractors, and the NCEA is what extends that protection to the external requester as well. And so the intent anyway, of course, I'd have your lawyer read through the agreement to be sure they agree, but the intent is to protect all of your confidential information and to make it clear that NIAID does not have any claim or the contractor have any claim to your intellectual property in the process of running these assays for you.
Yeah, I guess the intent of the question of that person was mostly to be reassured that NIAID is never going to share any kind of data with anybody that the company or the submitter does not want to share data with.
Yeah, okay, no, yeah, absolutely. So we will, transmit only the information that the contractor needs to run the assays. And as I said, that agreement keeps that information and those data in strict confidence. Outside of that, we do not, publicize or comment on any of the services that we're providing to people, so that stays confidential.
Thank you, Anne. Somebody here is asking if the slide deck is going to be available. The recording of the entire webinar will soon be available on the site, and you could see it as many times as you want. So another question here is just missing because we have a lot of questions and the time is starting to run out. So I guess you already answered that question. Somebody was asking if there was any kind of legal trade-offs if the company uses the preclinical services, so in case of royalties or something, and the answer is definitely not. Just so you don't have your IP compromised at all. Somebody's asking if NIAID offers any kind of biofilm in vitro testing.
Yeah, so we don't have an assay contract set up in place for doing that. I think it would be within the scope and capabilities of our contractors. So it would be something we could discuss possibly setting up. We don't have it in place right now, though.
Okay. Somebody's asking if the chemical synthesis is only limited to antimicrobial compounds, or if there is similar services in other NIH institutes that also provides these kind of services for molecules that maybe have different activities.
Right, I believe there are similar services in some of the various institutes. The ones that I described, have to be within the mission of NIAID, so would focus on the infectious disease space. So you'd have to check in with the other institutes on what exactly is available for other indications.
Thank you, Anne. Here is a question for Dan. Somebody's asking if you could share very briefly, because we don't have a lot of time, how did you start the company? So how did you manage to get your first grant, and a bit of the timelines until the company took off the ground?
Yeah, so we actually started the company on an R01 grant, and then but you have to allow that it's going to be roughly on the order of, I would say, 1 year from the time that you actually submit the grants, go up to 1 year to the time that you actually get the award. So again, it's the mantra of planning ahead. Once you have that first grant, then you can begin to, then you have the opportunity, I think, to begin to explore some of the other funding mechanisms and try to create some runway for the organization. But there is roughly a year lead time to getting funding from the first grant application, assuming that the grant application is actually successful.
Thanks, Dan. Here is also somebody asking, I guess this has been mentioned a couple of times, but what is the typical time needed for the review of the proposals? Will this depend on the type of study, if this is for a non-clinical, say, non-complicated study versus something more complicated, or on the clinical side, or something that involves GLP or GMP?
Yeah, no, exactly. I think you put your finger on the real variables. So, things that are like MIC testing, quick turnaround, ADME, the smaller studies, we try to set up contracts, including, some of the animal model work. We try to set up contracts that are, preloaded, in place, ready to go. So the turnaround time, once you're approved, can be pretty quick because we already have a contract in place. For things like, synthesis, scale up, that's a specialty and specific to the molecule you're asking about. And so we have to set up a new task order for putting that in place. And so that lead time's a bit longer. And again, for something like a very large scale manufacturing or clinical trial or other big ticket item, it would take a few more weeks of internal approval discussions than say an MIC test, which we can turn around much more quickly. So it kind of ramps up to a few weeks with approval to turn around out to a few months, depending on whether a new contract needs to be put in place for running the study.
Thanks, Anne. Somebody's asking also if is part of the preclinical services, for example, if somebody wants to synthesize a radioactive compound or a deuterated standard for analytical purposes, could that be synthesized to NIAID?
Yeah, that kind of work is in scope, and as I said before, the more familiarity you are with synthetic routes and experience you have with the specific methodologies, the better, and the faster and the more confident we would be in being able to turn around that kind of request. But generally speaking, that does fall, radio-labeled molecules do fall within the scope of our synthesis contract.
Thank you, Anne. Yeah, no, that one we already answered. So another question I have here is, will NIAID, for example, help to evaluate FDA-approved drugs that could be used as a combination therapy, or that will then, say, potentiate activity of antimicrobials?
I guess I'm not sure I understand the question. So they're asking, does that fall within the scope of things?
Exactly, they are asking if that will fall into the scope of the AMR area.
Yeah, so I think it depends on the specific funding announcement. Sometimes our funding announcements are geared, specifically to new chemical entities. Others are not. Like, for instance, the program announcement I think I mentioned is specifically looking for existing drugs and new combinations and grant applications to investigate that type of work. Whereas some of the other announcements are, like I said, focused maybe more on new chemical entities. So you kind of have to read the fine print of the particular announcement. For the preclinical services, I think it's just a matter of the evaluation of and justification of what you're asking for. It's certainly open. We're open to testing existing approved compounds or products, depending on what the need and the request and the justification is.
And one more question: does PCS offers formulation development?
Yes, it does, the full gamut of that. So whether it's therapeutics or biotherapeutics, that kind of work is in scope.
Thank you, Anne. So we have run out of time now. We received more questions than we could answer, and we thank all the participants for their active role in this. We will work with Anne and Dan to respond to the remaining questions, and we will update you about that in our follow-up emails. So this essentially, we have come now to the end of our webinar, and I would like to thank Anne and Dan and Dennis and Steve for their time, for sharing their expertise with our audience. And I would also like to thank the online participants for joining the webinar and for actively contributing to this discussion. If you have further questions, feel free to get in touch with us or with Anne or Dan directly. Again, make sure to join us again for our upcoming webinars. On March 28, we will have another webinar with David Schleis about development of antibacterial drug enhancer combinations and non-traditional products. And we will have regulatory experts participating in that seminar. Ian Friedland, Sumati Nambiar from the FDA, and Mayor Powell from the Medicines and Healthcare Product Regulatory Agency, MHRA in London, will be part of that webinar. And also on April 9th, we will have a webinar about mining chemical libraries for new antibacterials with Cara Balibar from the Merck Research Laboratories. I hope you found this webinar useful. We would appreciate to receive your feedback in the survey that we'll launch upon closing the webinar or by email. And I hope to see many of you again for our future webinars. Goodbye and good evening or good afternoon, depending on where you are. Thank you so much.